Hello. So actually, I have one of my yoga students in this room right now. I'm not going to call him out though, but people in the Forestry School do yoga. So my name is Brittany Barnes, and I am a research coordinator at the Warnell School of Forestry and Natural Resources. My boss is Dr. Kamal Gandhi, and she was actually the one who was invited to give this talk today, but she had to be in Colorado. So I got really lucky and got to come to Jekyll, and I have been working on this project for 4 years, and I have actually been one of the 2 people that have been in the ground doing all the field work, all the lab work for the past 4 years. So it's a project that I know fairly well. And it has been a multi-agency project. We have so many collaborators, so many people have been working with us on this. And the title of my talk today is The Interactions Between Abiotic and Biotic Factors Affecting Southern Pine Health. So when I give these talks, I generally give them to entomology people, and so I always have a slide like this in there, because a lot of people don't know how important forestry is to the Southeast and also to Georgia, but you guys know this, but I'm just going to emphasize it because it makes us feel really great. It is a multi-billion-dollar industry every single year. There's no wonder why we are considered the wood basket of the world. But we do have different factors that cause our trees to go through stress. We have different abiotic and biotic factors. Those are anything from drought to flooding to windstorms to wildfires. And then our biotic factors are normally and mainly our insects and our disease outbreaks. We have an interesting land use here in the Southeast. It used to be primarily here, especially in Georgia, longleaf pine. But then about 200 years ago, we started to tear down or cut down those trees and put in agricultural lands. And when you do that, you do deplete the nutrients of the soil, and you also cause erosion. So we do have that issue that we're always working with. We also used to have a really healthy wildfire regimen here in the Southeast, about a 1 to 3 year cycle. And with suppression, we do not have that anymore. And then, of course, urbanization has come and depleted some of our forests as well. So with all of these different factors that are involved, there's no reason to believe why our trees aren't as resilient to insects and disease outbreaks. So a little bit of historical dieback of southern pines. In about 1950s in Alabama, there started to be some trees that they started to see were dying. But there wasn't this one cause, this one reason for why these trees seem to be declining or dying back. And so this trend continued, and it continued in Louisiana and Alabama, Georgia, and South Carolina. So we've always been trying to figure out, what is going on there. There has always been different abiotic and different biotic factors, but there hasn't just been that one maybe cause that we can figure out. So this term, southern pine decline, started to appear. And normally when you have a decline, and there are declines throughout the country in different ecosystems, but when you have a decline, you have 3 factors. You have inciting factors, you have contributing factors, and you have predisposing, inciting, and contributing factors. Well, also with declines, what you see is that these declines are very widespread, and they're throughout an entire stand, and they actually move to the next stand. So with these trees that we have seen, we haven't really seen this. So this decline word was a really interesting one for us to start to read in literature. The literature started coming out about 10 years ago. And in this literature, there was always talk about this insect-fungus interaction. Now these insects, these weevils here, they are root-feeding rhizophagous beetles. They are secondary colonizers of trees. So when your tree is starting to get stressed and is starting to die back, these insects do come in and they help with the decomposition. That is their main goal. That is what they are here to do. And when they come into your sand, they do vector this fungus called leptographium. So you might see this leptographium fungus along with these beetles when you start to see your trees dying. Well, the University of Georgia and the Forest Service want to take a closer look to the southern pine decline that we're reading in literature, because it didn't really make sense to call this a decline. So we had some questions surrounding this that we wanted to really answer. our first question was, do these root-feeding insects and associated pathogens colonize healthy trees? We wanted to know, well, what is the extent and the severity of this pine health issue that we've been reading about? And then also, do other abiotic and biotic factors contribute to pine health issues or affect stand susceptibility? So for the first project that we did, we focused on this first question. Do these insects, these beetles, and their fungus, Leptographium, colonize healthy trees? So we set up this pretty large-scale study close to Athens, Georgia. In the fall of 2012, spring of 2013, and fall of 2013, we did 3 separate studies where we had 3 different treatment groups. We had a control where we had 4 trees that were perfectly healthy, they were unstressed, and we put a funnel trap, we put bread pan traps, and we put pitfall traps around these 4 trees. And we had 7 different replicates of this treatment. And then we had a second treatment where we wanted to mimic moderately stressed trees. So we had 4 trees, and then we didn't do anything to them, but we baited the traps, the funnel traps, the bread pan, and the pitfall traps with lures. And these lures had ethanol and turpentine, and those are terpenes that are released from trees who are going through a little bit of stress. So those were our moderately stressed trees. And then for our really stressed trees, we girdled the 4 trees and then put the traps around those 4 trees. So we have this really stressful environment. The trees were actually dying. So, well, did we get beetles? And this is just showing you that there's a lot of red here. And those red is the girdle. The red is the trees that were dying. There's very little baited, and there's hardly any control. So what we found from here was that these beetles probably don't colonize healthy trees, which is what's in the literature and what's been known, but this study was able to give us a little bit more concrete evidence that they're not coming into our healthy stands. So then we wanted to ask the question, or answer the question, what is the extent and the severity of pine health issues? Oops. So I'm not sure if any of you guys have actually read this paper, but it's a really lovely paper written by Dr. David Coyle and a bunch of his colleagues, where they looked at Forest Service FIA data in the Southeast. And these FIA data is taken about every 5 years. It's definitely every 5 years they go to each individual plot within the Southeast. Sometimes they go more recent than, or more than 5 years. But they found 5,500 plots that they were able to analyze to see how does the trees look? Are they growing or are they dying? Because whenever all these plots, DBHs are taken, heights are taken, and when the FIA folks see that something might be dying or there might be something happening, they actually write down the reason. So they'll either write down insects or disease or weather. And so out of 5,500, only 181 of these plots, so only about 3% of these plots showed any kind of negative net growth, which means there was very little trees that showed that they were dying. And these black triangles are the ones that show that negative net growth. And the map into the left corner is showing the Georgia and Alabama, because that's where we're really focusing with this pine decline project. And if we look just a little bit closer at that, we see what was contributing to this net growth. And out of all 181 plots that showed negative net growth, only 1 of them had no reason. But then when you go back and look at that one plot, they were 90-year-old trees. So that is a pretty good reason. When we look at this, we kind of see that it doesn't look to be a regional scale. When you look at them, there's not just one area that's just completely devastated, and especially with only 3% out of all those plots. So for us, this kind of showed that maybe it's more of a stand-level issue, stand-level problem. So the third question is, do other abiotic and biotic factors contribute to pine health issues or affect stand susceptibility? So for this one, this was our big 3-year-long study, where we went into 37 different sites in Alabama and also Georgia. We had 13 healthy and 24 unhealthy sites. And as you can see here in Alabama, all of our sites were in national forest, and all the unhealthy sites were already sites that were deemed southern pine decline sites. Whereas in Georgia, we worked with local state agencies to find our plots, and they were the ones who told us that these stands maybe looked a little unhealthy or there was some kind of mortality going on, but the issue, we weren't really sure why. And they were not only in national forests, so they were also in other land. So our sampling design, like I said, 37 sites. We also did the FIA style data analysis, where you go into a site and you establish 3 plots. From each plot, you establish 4 subplots. So that's 111 plots and 444 subplots. Now, within each subplot, we measured, with a 10-meter radius was our subplot, we measured the DBHs of every single hardwood, every single pine, every single dead hardwood, and every single dead pine. We chose 3 trees at random to take root samples from, to take core sample, to take cores, to take height, to do crown class ratio, overall health. We analyzed these trees for any kind of cankers, mechanical damage, insects, you name it. So, and also at each subplot, we also took slope, aspect, and soil samples. That made us measure over 11,000 trees and 1,300 pages of Excel data. So if you've been wondering where our data has been, it's been in 1,300 pages that we've had to go through. And we're still fine-tuning our analyzations, but I do have some p-values for you today, so that's exciting. In order for us to do our fungal sampling, we wanted to look obviously at leptographium, which again is that fungus that is vectored by the root-feeding weevils. But we also want to look at heterobasidium or anosum root rot. In order to do this, we took 3 root samples from each tree within the subplot. So from each subplot, we took 3 trees, so we had 9 root samples from each subplot. We took a large root, a medium root, and a small root. We brought all those roots back into our lab, where we then cut up each root and we placed them into a media. And this media was specific to try to grow leptographium. And then we also cut a chunk of the wood and put it into a plastic bag with a wet paper towel for heterobasidium to grow. Now, when we saw some fungus growing, we then put the fungus under a microscope and we looked at it. And we looked to see if we saw any leptographium spores or any heterobasidium spores. Now, if we did see it, then we took a very tiny, tiny metal point and we found that one little spore, and we put that point into the spore, and then we put it into another plate of media, and we grew a pure culture. And this is a pure culture of leptographium. And we wanted to make sure we did that so we were 100% sure of what we were seeing, if we were seeing leptographium, if we were seeing heterobasidium. So, what did we find? Well, this is a picture that was taken back in 2012 when my professor received a phone call from somebody saying, I have pine decline in my site. And so she and some other colleagues went to this site to take some root samples and kind of see what we could figure out. And I don't know if you can see, but there's some people in that picture. And what she saw automatically was that there was a lot of hardwood growing right there. And so for my first statistic here, I want to show you that it was significant that the higher the hardwood basal area was higher in unhealthy sites than healthy sites. And yes, that sounds really like common, and you know this, but this is what we saw, and you know this, so we know what to do about that. What we didn't find, though, was any difference in leptographium in all of these sites. And we actually saw a little bit more leptographium in healthy sites than unhealthy sites, but there was no significant difference. We did find, though, that there was a significant difference in heterobasidium or anosum root rot, where there was more heterobasidium in your unhealthy sites, your symptomatic sites, your sites that are showing decline versus your healthy sites. So because of that, I kind of wanted to go a little bit over heterobasidium. It has a lot of different names, but it does affect loblolly, longleaf, pitch, shortleaf / Virginia white pine. It is found in pine plantations that just had a thinning, and it's usually found on well-drained sandy soils. It causes a reduced tree growth, mortality, and because of mortality, you can sometimes see an increase in bark beetle attacks. And how heterobasidium works is that when you thin, you have this fresh stump. Well, the spores go into that fresh stump, they germinate, and then the fungus grows through the root system. And now the spores are in the root systems. And our roots are all integrated down below. And so when a healthy tree touches the root system of an unhealthy tree or a tree that just had heterobasidium, then it can spread. And that's kind of how it starts to spread. A little risk factor for you. This is a soil map, risk factor map. So sandy loam soils, about 65% sand, are a little higher risk. And also losses are higher on former ag land than on forest soils. And this map in the red is your highest risk, brown is moderate, and tan is lowest. And a little bit of strategies you can do for management. There are some treatments that you can use after thinnings. Summer thinnings are encouraged if you're south of the 34-degree north latitude line. And as you can see in this picture, that's just a little bit above Atlanta. So most of y'all's lands is most likely south of that line. And the reason for the summer thinning is that it gets so hot here. So when that spore wants to germinate on the stump, it dies because it's so hot. So that's a really big benefit of heat. They also say wider spacing between trees to delay or reduce the number of thinnings, and then also select species that are less susceptible, like longleaf pines on those high hazard soils. So in conclusion of those studies, what we saw was that these beetles are associated with stressed and not unstressed trees, so they're usually in unhealthy or symptomatic stands. It seems to not be a region-specific issue, but maybe more of a site-specific issue. There was no relationship in Leptographium fungus, but there was a relationship in Heterobasidium. And so you need to consider all your abiotic and biotic factors along with your proper management when it comes to your pine health. So we also wanted to know, what do you all think? What do y'all think about southern pine decline? What do y'all think? How are your forests doing? So we also did a survey, and I bet some of you guys maybe took it, and if you did, thank you very much. But we wanted to look at three main things. We wanted to assess the perceptions of forest landowners and managers regarding southern pine health. We wanted to determine what, if any, management landowners and managers use. And we also wanted to figure out the best way to communicate forest health information to landowners and managers. So our method was that we acquired addresses from state forestry agencies in Georgia, Florida, and South Carolina. We ended up emailing and mailing 4,597 forest landowners or managers. And in this survey, there was three components. The first component was a section just on general health of your pine stands. The second section was on knowledge of southern pine decline. And the third was just demographics. So we had 1,287 usable responses. The non-usable responses went like this: go Gators. And then there was no answers. So got many of those. But we did have a 28% overall response rate, which in survey world is really good. So we were actually very happy with that. 80% of those who answered were males, 90% white, and around an average of 64 years old. So I'm just going to go through a few of the what we found from that survey. Most respondents have not seen dead or dying trees within the past year. Great. It looks like the majority of respondents appear to have not seen an increase of dead or dying trees on their property. Great. And if you did, most people believe it was either insects or drought, which is a very common, that's what most people think, and it's not, that is wrong by any means. And also, people prefer to get their information from handouts and brochures from universities or state agencies. And the internet is up there, but I do believe that because of the demographics we answered it, I think the internet is probably increasing as well. And also, most landowners are willing to use acceptable stand management techniques. Yay. And then this one was funny to me, but 71% of people have never even heard of southern pine decline. So in that section, nobody even answered because they have never even heard of it. So what did we find? Well, we found that southern pine stands seem to be overall healthy, with landowners not noticing dead or dying trees within the year or noticing an increase in tree mortality. We saw that respondents believe drought or insects are the reason. Landowners and managers are willing to get information from the universities and the federal sources and state sources through handouts and brochures. And landowners are willing to do proper sand management. And southern pine decline, maybe not as well known as we think. But I want to end with this, and this is information from a fellow forester, but proper management is key. Do what you do best. Keep doing it. Things are doing really well. I believe that Amanda today with Forest said that 90 out of all of the survey they did, 90% was survival rate. So that's really good. We're doing really well. But manage your forest in a method that will keep the forest viable and sustainable throughout the entire rotation. Thin your stands when they need to be thinned. Prescribe burn in order to keep your understory manageable. Proper species on proper sites. And walk through your forest on a regular basis, because if you see some insects or you see disease, then you can manage quickly and properly. So just be proactive. And with that, I do want to acknowledge a lot of people who have helped me with this project. And then I also want to end with resources, because we have a lovely team of forest health specialists here in the state of Georgia that are here for you all. So all these people are willing to answer any questions that you might have. And one of them is about to give a lovely talk right after me. So thank you.